All right, it's 2 o'clock, so we're going to go ahead and get started. If you haven't already, please go ahead and mute your microphone and turn off your camera. That'll just make the system work better. And if you have a question, just unmute it and ask away. That's what we want you to do. We want you to ask questions. So let us know if you come up with any questions at all. What we're going to be dealing with today is some stuff about statistics and also about experimental design. I think the first question you're going to get on the AP exam is probably going to be pretty broad. And it's going to have probably much to do with some kind of experiment setup and an evaluation of that experiment. Whether we're doing stats or not, we don't know. But I would guess there might be a stats component to that question. I don't think you'll have to draw a graph because that might be kind of hard to do on the computer for some people. And also, they're not sure if you have graph paper at home. So I'm guessing it's probably going to be a graph interpretation kind of question, or maybe a data table interpretation. But for sure, there'll be some kind of experimental design and data interpretation component to that question. So what I'm going to do today is I'm going to look at 3 questions related to stats and related to experimental design. We're going to start with this question. So if you guys will take just a minute and read that question, we'll get started here momentarily. All right, so some of you probably seen this question before. Trichomes are these little hairy-like growths on some plants. Scientists think they're probably about preventing herbivores from eating the leaves. Not 100% sure their function, but that's what they think they're probably about. So in this particular setup, a scientist is investigating the variation in how many trichomes are on these plants, the trichome density. So they've given you 3 setups, 3 populations that they've sampled from, and they've looked at 6 plants from each of these populations. Now notice they've already done the math for you. They calculated the mean trichome density measured in number of trichomes per square centimeter for each population. They also calculated the standard error of the mean for each group. Now realize that in the real world, you're never going to get a standard error of the mean of exactly 1. And you're not ever going to get the same standard error of the mean for 3 different populations. But here they gave you that number just to make what we're about to do a little bit easier. Now, the first thing they ask us to do is to do a graph. Now, remember, they're probably not going to ask you to do a graph on this exam. On a normal AP exam, yes, they're going to ask you to do a graph. This one's strange. But I'm going to go ahead and free draw this. I don't think I have a piece of graph paper handy. Actually, I already did it here. So let's look at what I did computerized. So if we're graphing means, probably the easiest type of graph and the most appropriate graph is just to do a bar chart or a histogram. Remember that we're going to do a graph with X and Y. And what we always want to put on our Y-axis is the dependent variable, which in this case is going to be trichome density. And what we're going to put on the X-axis is our independent variable, which in this case, not really the independent variable experiment, but it's going to be the population number. So we're going to put pop number against trichome density. Now, if you were asked to do a graph, make sure that you also include the units and that you clearly label both axes. Notice what they're asking you to do here. And I can see I'm asking you a question about a pre-made graph on this one. So we want to illustrate the sample means to within a 95% confidence interval, or go plus or minus 2 standard errors of the mean. So here's my graph. Notice population 1. Our mean on that group was 9. I'm going to go ahead and label that as X bar equals 9. That's right here. For our second group, our mean was 11. And our last group, our mean was 14. You don't have to label these if you're drawing the graph, but I'm going to label them just so I have that handy. So next thing they ask us to do is to create these 95% intervals. So here's what we're doing. Let's go back to the data table for just a minute. We've got these 3 groups of plants. Maybe let's think of it as 3 fields of plants. There's probably thousands of plants in each field. We're not measuring or counting the number of trichomes on each one of those plants. We randomly sampled 6 plants out of each field. And what we're wanting to do is to draw some conclusions about the whole group from this population. So we're doing what we call inferential statistics. So we want to use a sample mean. We've got a 6 plant sample from each group. We want to use that small sample to make predictions about the whole population. So let's go back to our graph and talk about how we do that. So to create a 95% confidence interval, you're going to start at your mean, and you're going to go up from that mean 2 standard errors of the mean. Remember, our standard error of the mean for population 1 was 1. So we're starting here at 9. That means we need to go up 2 ones. We need to go up to 11. And we'll draw our error bar like so, just a little bar. And we're going to go with a vertical line and connect that directly to the mean, like so. To finish up our 95% confidence interval, we also need to go down 2 standard errors of the mean from the mean. So again, our mean is at 9. We're going to go down 2. So we'll go down to 7. We'll draw a horizontal line like so. That's a terrible horizontal line. Let me try that again. And we'll connect that with a vertical bar to our mean. That's a 95% confidence interval. And what that means is from a sample mean of 9, we can predict with 95% confidence that the mean of the whole population for population 1 is somewhere between 11, this upper boundary, and 7, this lower boundary. So we're predicting from the sample mean, that small sample mean with a mean of 9, that the mean of the whole population of population 1, there's a 95% chance that the mean of that whole population lies between 7 and 11. Let's do the same thing for population 2. So population 2's mean was at 11. So to create a 95% confidence interval, we're going to go up 2 standard errors of the mean from that. So that's going to put us up here at 13. We'll draw down our vertical line to our mean like so. We're also going to go down 2 standard errors of the mean. So we'll go down to 9. Connect that with a vertical like so. So for population 2, we can be 95% sure that the mean of the whole population is somewhere between 9 and 13. So 95% sure that the mean of population 2 is between 9 and 13. And finally, for population 3, the mean there was 14. To create that 95% confidence interval, we're going to go up 2 standard errors of the mean. So from 14 up to 16. So remember, the standard error was 1. We're going to go down also 2 standard errors. So down 2 from 14 puts us at 12. So for population 3, we can be 95% sure that the mean of the whole population is somewhere between 12 and 16. So that's the 95% confidence interval for population 3. For population 2, it's somewhere between 9 and 13. And for population number 1 was between 7 and 11. So keeping that in mind, let's go back and look at what the question was asking us on this one. So we did part A. We drew the graph. We've got the 95% confidence intervals. Part B is probably the kind of question you're going to see on this year's AP exam. Based on the sample means and the standard errors of the means, identify the two populations that are most likely to have statistically significant differences in mean trichome densities. And there's that word we're going to see in all kinds of AP questions. Justify your response. Back it up with some data. So which two are most likely to be different? To determine that, you want to look at the 95% confidence intervals. Notice that population 1 and population 2 overlap. Their confidence intervals do right in here. That means there's a chance that the actual mean of population 1 and 2 are the same, and it could fall somewhere in this circled region. Notice that also 2 and 3 overlap right in here. That means there's a chance, a pretty high probability, that the mean for population 2 and population 3 is the same. The two that don't overlap are 1 and 3. There's no overlap between this confidence interval and this one. So if we go back to the question, first of all, we're going to identify. So we might say the two populations that are most likely to have statistically significant differences in mean stem trichome densities are populations 1 and 3. And then the justification is going to be because their confidence intervals do not overlap. Their 95% confidence intervals do not overlap. And really, that one sentence is all it takes to answer part B. You don't really need to say any more than that. The identify part, populations 1 and 3 are the two that are most likely to have statistically significant differences in mean stem trichome densities because their 95% confidence intervals don't overlap. You might also add in that the confidence intervals for 1 and 2 do overlap, and the confidence intervals for 2 and 3 overlap. But the confidence intervals for 1 and 3 don't overlap. Therefore, they are likely to be statistically significant. The differences are likely to be statistically significant. All right, so that's it for B. Now, I said that also on the exam, you're probably going to get some questions about experimental design, experiment setup, that kind of thing. So I want you guys to take a minute and read part C and just sketch down what you think the answer is, or just get it in your head, either one. But let's take about 2 minutes, look at part C. Try to come up with an answer yourself. All right, so again, pay attention to the verbs here. So first of all, we want to describe the independent and dependent variables and a controlled treatment. So 3 things. For an experiment to test the hypothesis that higher trichome density in plants is selected for in the presence of herbivores. Essentially, they're saying that more trichomes means the plants don't get eaten as much, is essentially what that question is saying. So that's part 1. Second verb, identify an appropriate duration of the experiment to ensure that natural selection is measured. And third verb, predict the experimental results that would support this hypothesis. So I would just do this in order. Remember, by the way, you want to answer these parts of the question, write down part A, write your answer to part A. Write down part B, write your answer to part B. Now we're on C. So write down part C. And I would say in this experiment, my independent variable is, think about what an independent variable is. The independent variable is our hypothesis is that higher trichome density is selected for in the presence of herbivores. So I think the most logical independent variable here is trichome density. So I'm just going to abbreviate. Independent variable is going to be trichome density. So in a setup, I might have 3 groups of plants. One with a low trichome density, one with a medium, and one with a high trichome density. Then I might expose all of those plants to the same herbivore. Turn the herbivore loose wherever my plants are growing. 3 different setups. My dependent variable could be a lot of different things, but remember, it's the thing that's impacted by the independent variable. And I could look at plant survival. How many of the plants survive after being exposed to the predator? I could look at the amount of leaf eaten. A lot of different things I could look at. Anything that has to do with plant survival, plants not being eaten, any variables that look at that. The last part of this, or the second part of this question, identify an appropriate duration of the experiment to ensure that natural selection is measured. Now, the AP exam this year is not really dealing with evolution. That unit is removed from the test. But what you've got to realize is that individuals can't evolve. So the only way this experiment can measure if natural selection happened or not is if it happens over multiple generations. That's the thing they're looking for you to talk about here. So I would say that an appropriate duration of this experiment would be several generations of this particular plant. That's what they've got to know. They want you to know that it's not the individual that evolves, it's the population that evolves. So you've got to say something about multiple generations. And then finally, predict the experimental results that would support your hypothesis. Remember, our hypothesis is that higher trichome density in plants is selected for in the presence of herbivores. So really what we're saying here is that plants with more trichomes are going to outlive and out-reproduce plants with lower trichome densities. That's essentially would be good experimental evidence. Let's go to the grading rubric that was used for this exam. So here's how the graders actually graded the test. You could get 3 points for part A. So that was for labeling and scaling your graph, putting in the error bars, creating the confidence intervals. 3 points for doing that. For part B, first of all, identifying the 2 populations that were most likely to be different. That's number 1 and 3. And then the justification was essentially that the error bars for those 2 groups don't overlap. 2 points on part B. And then on part C, 1 point for identifying the variable. And notice the way that I did mine was the bottom way they did it here. So the trichome density. The dependent variable could be reproductive success. The control, and I didn't mention the control in mine. So a control would be plants with low trichome density or plants with no trichomes, might even be better. The duration, multiple generations. And then the size of the population with higher trichomes is bigger than the population with lower numbers of trichomes. So it's got to be about reproductive success, because we're talking evolution, and it's got to happen over generations. One individual can't evolve. The population evolves. That's what they're wanting you to talk about. So that's the first FRQ for today. Are there questions I can answer for you guys about this one? All right. If not, let's go on and go to the next one. So everybody take a minute and read this. We're going to look pretty closely at part A. We're not going to be as concerned about parts B and C because they really deal with evolution. But I do want you to look very closely at A. So go ahead and read it, and we'll talk about it here in just a minute. All right, so we're looking at guppies here, which are type of fish. And they exhibit genetically determined spots. And these are only in the male guppies. Juvenile and adult females don't have spots. So in a study of selection, male and female guppies from genetically diverse populations were collected from different streams, placed together in an isolated environment containing no predators. So we're starting with no predators in the pond. The study population was maintained for several generations. Notice generations. Evolution happens over generations. In an isolated pond before being separated into two groups. One group was moved to an artificial pond containing a predator, while a second was moved to a pond containing no predators. The two groups went through several generations, there it is again, in their new environments. At different times during the experiment, the mean number of spots per adult male guppy was determined as shown in the graph below. Vertical bars in the figure represent two standard errors of the mean. What I want you to remember about that standard error of the mean is it's derived from the standard deviation. And that standard deviation, and by the way, it's essentially, this is the formula. It's on your formula sheet. It's the standard deviation divided by the square root of the sample size. Standard deviation, though, is a measurement of variation. Data sets with a large standard deviation have more variation. Data sets with a small standard deviation have less variation. The same can be said for the standard error of the mean. Bigger standard error of the mean means more variation. Smaller standard error of the mean means less variation. So keeping that in mind, let's look at question A. Describe the change in genetic variation in the population between 0 and 6 months and provide a reasoning for your description based on the means and the standard error of the mean. So what we notice at 0 months, the mean number of spots is 10, but our 95% confidence interval goes almost all the way down to 8 and all the way up to 12. Hopefully that's telling you that the standard error of the mean there is close to 1, right? Because we go up from 10 to 11 for one standard error and up to 12 for 2. So SEM here is approximately 1. But notice when we get to 6 months, which is right here, the mean number of spots is about 12, almost 12. But notice how much smaller the 95% confidence interval is. It's less than 1/2, less than probably a fourth. SEM is maybe 0.25. When the SEM is smaller, that means that the standard deviation was also smaller. And if the standard deviation is smaller, that means there's less variation. So if you look at the question, describe the change in genetic variation in the population from 0 to 6 months. Here there was lots of variation because of the big SEM and big standard deviation. Here there wasn't much genetic variation because of the small SEM, small standard deviation. So essentially how you want to answer this question is you want to say that the amount of genetic variation drastically decreased between 0 and 6 months. And the reasoning you're going to provide is because the 95% confidence intervals and therefore the standard error of the mean was much higher at 0 than it was at 6 months. So let's look at the grading rubric on this. So here's what the graders used to grade it. 1 point for saying genetic variation is decreasing. 1 point for saying SEM gets smaller. Now we'll go ahead and quickly look at B and C, but we're not going to spend much time on those because they're about evolution. B, propose one type of mating behavior that could have resulted in the observed change in the number of spots per adult male guppy between 6 and 20 months in the absence of a predator. So if you look at your graph, predator absent, solid line, that's this part. And notice that the number of spots, the mean number of spots, increases from close to 12 up to a little bit more than 13. Mean number of spots goes up. And we're trying to describe a mating behavior that would have resulted in that change. What the spots are about is attraction of a mate. This is what we call sexual selection. So my answer here would be that sexual selection is going on, that females prefer males with more spots. Therefore, males with more spots mate more often and reproduce more babies. So the genes for lots of spots increases faster than the genes for not many spots. Again, looking at the grading rubric, sexual selection for individuals with more spots, or random mating behavior resulted in increased number of spots by chance. Either one could happen. This is probably not nearly as likely as this one. That's the answer they're looking for. 1 point for that. And then the second one, propose an evolutionary mechanism that best explains the change in average number of spots between 6 and 20 months in the presence of the predator. That's this part of the graph. And again, we're going from a mean number of spots of close to 12 all the way down to about 9.5 here. So the mean number of spots is dropping very quickly. Here's the thing with spots on a fish. The spots are meant to say, hey, here I am. And they're saying, hey, here I am to the females. But at the same time they're being seen by the females, that's also making them more easily spotted by predators. So when a predator is present, males with lots of spots are being seen faster. Therefore, they're being eaten faster. Therefore, they're not able to reproduce as much. So the genes for lots of spots starts to decrease in the presence of a predator. What I just described is natural selection. So the mechanism they're looking for is natural selection because the mean number of spots is dropping because males with lots of spots are essentially getting killed and they're not able to reproduce. So if we look at the grading rubric for that, natural selection in context, and directional selection is a kind of natural selection for individuals with fewer spots. They also mentioned genetic drift here, which is sort of saying it's random. I guess that could be the answer. That's not nearly as likely as one of these top three. All right, that's it for question 2. And that's about as much as I'm going to say about confidence intervals and SEMs. I want to go ahead and look at one additional question for today. And this is what I think a question on the exam really might look like. So take a minute and read through everything you can see on the screen, and we'll answer this question here momentarily. All right, so these choice chambers labs are a very common lab that you'll see done in AP Bio classes. A lot of you probably did a version of this lab with pill bugs, with essentially the same setup you see in this picture. Here we have glucose-soaked cotton on one side and dry cotton on the other. We put some flies. Let's say 60 flies are introduced into the middle in here. And then we're asked to figure out what's going to happen. So part A, predict the distribution of flies in the chamber after 10 minutes and justify your prediction. Most of you are probably going to predict that most of the flies are going to move to this side. And that would be a good answer. Most of the flies will move to the side with the glucose-soaked cotton. That's a prediction. Your justification would be that the flies feed on glucose. Glucose is a food source. So let's go to the grading rubric and see what it looked like for this. So 1 point for predicting, 1 point for justifying. And here they would take anything that's logical, but most people are going to predict they go to the glucose side because they feed on glucose. That's the obvious answer for this. Now let's look at part B. Propose one specific improvement to each of the following parts of the experimental design and explain how the modification will affect the experiment. The control. Now think about it. What's really the control in this experiment? What we really want to know, I guess, do they like glucose? That's really our independent variable. What our control really is here is this side. What's going on this side over here? And we use dry cotton. And I'm going to tell you that's not a good control. Because notice this is glucose-soaked cotton. Well, if it's soaked, and it tells us here it's a 10% glucose solution. So if a solution is 10% glucose, what's the other 90%? That means it's 90% water. So really on this side, we have two things going on. We have sugar and we have water. On this side, we don't have either one of those. So if all the plants go over to this side with the glucose-soaked cotton, we don't really know for sure in this experiment if they're going over there for the glucose, if they're going over there for the water, or if they're going over there for both of them. So a better setup on the experimental control would be to not make this not be dry cotton, make this be wet cotton. Include water on the experimental control side. Notice they also ask us to look at environmental factors. So let's go back to our rubric for a second. Here's the control. So the most common answer here, replace the dry cotton ball with a water-soaked cotton ball. And the explanation, that lets us know for sure that glucose is the thing the flies are attracted to and not water. They could also talk about, you could have also talked about, instead of that, keeping certain things constant, like the amount of light or the temperature, the duration, the importance of holding things constant. But I think this first line, this top line, is the way most people would go. Now, in terms of improving the environmental factors, they mentioned using different levels of glucose, different concentrations. So you could have multiple choice chambers set up. One might have a 10% glucose, one might have a 15, one might have a 20, one might have a 25, one might have a 30. So run different trials with different glucose concentrations. You could look at different temperatures, different light levels. You could vary the size and shape of the choice chambers. You could vary the duration, even times of days. Do you want to look at, is it just glucose they're looking for, or do other things play in? I think the first part of this, the control, was much easier to answer than the second part. Well, I really wanted to look at this question, though, was part C. The experiment described above is repeated with ripe bananas at one end and unripe at the other. Once again, the positions of the flies are observed and recorded every minute for 10 minutes. The positions of flies after 1 minute and after 10 minutes are shown in the table below. So here's our table. Perform a COSSquare test on the data for the 10-minute time point in the experiment. The 10-minute time point. So this set of data. Specify the null hypothesis that you are using, that you're testing, and enter the values from your calculations in the tables below. All right, so the null hypothesis. First of all, let's talk COSSquare. So a COSSquare is essentially a statistical tool that will allow you to compare an observed set of data. So a set of data that you got in your experiment to a set of data that you expected to get, what you thought should have happened. And we're going to base this on a null hypothesis. So remember, null means none, essentially. So a good null hypothesis for this experiment might be that that plants, or that fruit flies in this case, have no attraction to ripe bananas or unripe bananas. So therefore, the flies should be evenly distributed between the end with a ripe banana, the middle, and the end with the unripe banana. So essentially, that the presence of the banana has no effect on where the flies locate themselves. That's our null hypothesis. So now we want to test it. And they gave us this data table right here. So notice observed, end with ripe banana, 10 minutes, 45. The middle, we had 3 flies there, and the end with the unripe, we had 12. Well, if we add those up, 45 plus 3 plus 12, that's a total of 60 flies. Now, these expected numbers, these are based on our null hypothesis. And our null hypothesis was that the presence of the banana has no effect on where the flies locate themselves. Therefore, the flies should be located evenly in the 3 places. There's 3 places. There's 60 total flies. We're going to divide 60 by 3. Get 20. If the presence of the banana has no effect, those flies ought to be evenly distributed between our 3 places. If I was doing the math here, I would have a couple more columns in my table. So I've got OE. My next column would be O minus E, and then O minus E squared, and then finally O minus E squared over E. So O minus E is observed minus expected. So I'm going to go 45 minus 20, 25. 3 minus 20, negative 17. 12 minus 20, negative 8. Then I would square those. Let me get a calculator here. So first of all, we're going to go 25 squared. We're going to get 625. We're going to do negative 17 squared. Remember, 17 negative times negative is positive. So 17 times 17 is 289. And then negative 8 squared, negative times a negative is positive again, 64. Now we're going to go do O minus E squared, this column, divided by E, this column. So in the top row, it's going to be 625 divided by 20. 625 divided by 20. Struggling with my calculator here. Give me just a second. 625 divided by 20. So that's 31.25. Next, we're going to have the 289 divided by 20. So that's 14.45. And then finally, the 64 here divided by the 20 there. So that gives us 3.2. All right, so the COSSquare statistic, the formula for it, which is on your formula sheet, is equal to the summation of O minus E squared over E. This is the symbol for COSSquare. So that essentially is telling us we want to sum up this last row. So let's add up everything in that last row, and let's see, that's going to be 0, 1, 48.90, I think. Let me check my math there. Yep, 48.90. That's our COSSquare statistic. Now, by itself, that number doesn't mean much. We have to compare that to a standard value in the COSSquare table, which is given to you on your AP exam on your formula sheet. Let's see if I have that here. All right, I don't have the COSSquare table, so let me pull that up real quick. Give me just 1 second to pull up the COSSquare table. Bear with me for just a minute. I'm just going to pull up the formula sheet so that you can see it there. And by the way, I would make sure that I have a copy of this formula sheet printed out. I'd recommend you have a hard copy printed out and beside you when you start taking the exam. And if you don't have a copy, you can email me, matt@aplusala.org, or email your teacher, and they can send you a hard copy. This is what the formula sheet should look like. Here's the COSSquare part of the test right here. So to use the table, you first got to know degrees of freedom and the p-value to use. So let's go back to our data here for just a minute. Oops, I lost all my numbers, guys. Remember, our COSSquare value was 48.9. we had essentially three different places those flies could be: the end with the ripe banana, the middle, or the end with the unripe banana. Three possibilities. Our degrees of freedom are essentially one less than our total possibilities. So our total possibilities minus one. So we had three possibilities minus one. Our degrees of freedom are two. So now if we go back to the formula sheet for a second. So we're going to be working from 2 degrees of freedom. Typically, you use the p-value of 0.05, 5% probability level. So we want to look where the 0.05 row crosses with the 2 degrees of freedom column. And we end up with a value of 5.99. That value is called our critical value. That's the value from the chi-square table, the critical value. And here's really what you need to know. If chi-squared is greater than your critical value, that essentially means that you can reject the null hypothesis. Now remember, our null hypothesis was that the flies don't really care. The banana, either the ripe or the unripe banana, has no effect on where the flies locate themselves. We're rejecting that null hypothesis. So that's telling us that the bananas do have an effect. Essentially, we're showing that with statistics. If chi-square is greater than the critical value, you reject your null hypothesis. If, on the other hand, chi-square is less than the critical value, you fail to reject the null hypothesis. In our case, our chi-square is much bigger than our critical value. We're going to reject the null hypothesis, and that's essentially telling us that we've got some evidence that the presence of the bananas do affect where the flies locate themselves. So let's go back to the question for a second. So we did the chi-square test. We specified the null hypothesis that the bananas don't affect where the flies locate themselves. We entered the values into the table. Our null hypothesis is not supported, and our justification is chi-square is greater than the critical value. And then finally, propose a model that describes how environmental cues affect behavior of the flies in the choice chamber. Essentially, all they want you to say here is that the environmental cues act like a stimulus. And that stimulus is picked up by receptors in the brain or in the body. Routed to the brain. Processed in the brain. And then ultimately, that causes a response. So essentially what they wanted you to say is that a stimulus, in this case, the presence of the bananas, ultimately causes a response, which in this case is to move toward the ripe bananas. Stimulus response. That's what they wanted you to mention. So before we call it a day, I want us to look at one more question. I really think that this AP exam might be one of the easier ones we've had in a long time, because I think the questions are going to have to be pretty general. This is a very general question off of an AP exam from 6 or 7 years ago. It's on the new form of the exam. This is something I think questions might look like just from knowing how things are going to go here. So take just a minute and read this question, and it's as easy as it seems like it is. There's no trick here. So read parts A, B, and C, and then we'll talk about this one. I think number one might look something like this. So part A, pose one scientific question that the researchers were likely investigating with this experiment. Well, the independent variable in this experiment looks like it's ethanol, the concentration of ethanol, because we have no ethanol here. The water is the control. And then we have two different amounts of ethanol, 1% and 5%. So the question, it looks like what they're trying to ask is, what effect does the presence of ethanol or the concentration of ethanol have on the amount of urine produced in mice? And that's really all you need to say on A. So if we look at the rubric, propose one scientific question that researchers were likely investigating. How does alcohol consumption affect urine output in rats? Or how does alcohol consumption affect regulation of the kidney? Either one of those is okay. 1 point was awarded for that. Part B, state a hypothesis that could be tested to address the question you posed in part A. For part B, I don't think they care how you write the hypothesis. Some teachers want you to write if and then describe what you do to the independent variable. Then what happens to the dependent variable. Some just have you write it as a statement. I might write it, if mice are exposed to increasing amounts of alcohol, their urine output, the dependent variable, will increase with those increasing amounts. That I didn't say that very well, but essentially, alcohol consumption increases urine output in rats. That's essentially what we're looking at. You could say it in lots of different ways, but either way, more alcohol means more urine. Now, you also could have correctly said it the other way, that alcohol consumption reduces urine output. The data doesn't seem to back that up, but you could test whether or not this is true, because the thing they wanted was state the hypothesis that could be tested with this kind of setup. Either one's okay. And then finally, part C. Let's go back to our question here in part C. Using the data in the table, describe the effect of ethyl alcohol on urine production. So it looks like with no alcohol, this was the amount of urine production. With 1% alcohol, notice it increased. With 5%, it increased even more. So it looks like increased alcohol concentration causes an increase in urine production. And that's all they really needed. Alcohol consumption increases urine output. So you described the effect. Very, very general question. I think the questions on this year's exam are going to have to be pretty general, simply because you've got two questions that are grading a whole course. So I think the questions are going to be a little bit more difficult than this one, but I think it probably will be something similar. Very general questions that you just have to look at the data, answer the specific parts of the question that they're addressing to you. Those are the questions that I wanted to look at for today. Can I answer questions for anyone?